The University of Michigan Division of Gastroenterology and the Inflammatory Bowel Disease Program host the IBD Visiting Professor Lecture Series in Ann Arbor, Michigan. This series presents the latest in clinical research and basic science from IBD experts from around the world.
All right, well, good morning. First of all, let me thank Peter and really everyone for just an extraordinary visit. I was honored to be invited to be here, and you guys have been fabulous hosts, and I've been really impressed and enjoyed every minute I've had so far. So Peter did email me and said, please pick a topic that's new or different or something we haven't seen or that you haven't published quite yet. And so I had lots of things I hadn't published quite yet, but I was actually very interested in this topic. We've actually been working in this field now for a couple years, and the interest comes from an emerging understanding and appreciation for the importance of the microbiota in inflammatory bowel disease, which, as I'm going to show you in a moment, is not a new topic at all. But as people garner their enthusiasm for this topic and do some really intriguing and important science related to it, we have to start thinking a little bit more about what this means for translation into humans and doing good clinical trials. The reason I have a particular interest in this is not just because I focus on IBD, but also I spent an extra year and did a clinical ethics fellowship at the University of Chicago and had some interest in the clinical ethics of human subject research and protection of human subjects. So whenever something new like this comes along, it's a perfect opportunity to apply some of what we know about protecting human subjects and think about how we're going to apply some new technologies or new science. The picture, which a couple of people in the room definitely recognize, is the University of Chicago campus. The hospital is actually off the screen to the left. And for those who keep asking me, where exactly is the University of Chicago? Here's a picture for you. Downtown is obviously at the top of the screen. We're only 4.5 miles from downtown. So when you come to DDW, it's here in McCormick Place, and we're right down here. So this is our campus. It's very beautiful, and Hyde Park, where the university is, was the first suburb of Chicago. It used to be actually right next to downtown Chicago, and then after the Chicago fire, they moved the city downtown a little bit further away. So that's where I live, actually, and that's where we work. So I'm going to cover a broad range of topics, but they're all focused on a general understanding of how our appreciation of the gut flora applies to IBD and how we might try to modulate it to treat IBD. This isn't new, and in fact, if we go back in the history of studying IBD, I'm going to show you that there's actually quite an interesting historical perspective on all of this. So I'll cover the historical perspective. I'll talk to you a little bit more about why there's a good rationale for the microbiome playing an important role in patients with IBD, and not everybody. The heterogeneity of IBD is well appreciated, but understanding that there are some people in whom this is a particular issue is part of what we'll focus on today. And then I'll break down some of the discussion of how do we translate the science into human trials based on disease-related, microbiome-related, patient-related, and study design-related issues. So allow me to create a construct for you. This is something that I made up. So forgive me a bit, but it comes from my mentorship with someone who has perspective on the history of IBD, and that's my first GI mentor, Dr. Joe Kersner, who I met when he was 80 and is now 102 and still comes to work a half day a week. So I've really heard from him about where we've come and where we're going, and it's interesting to see how much things are coming full circle, and I'll show you that in a moment. So I've broken it down to 3 eras. The first one I call the era of the disease, which is when IBD emerged, and I call it also the era of desperation. We didn't understand it, we didn't know where it came from, and there were no good treatments. People died from this disease, people had substantial morbidity. Our treatment options were based on whatever we could get our hands on to try and prevent death, let alone focus on anything that we currently think of as being disease control or modified outcomes. Later in this era, there were really no effective medical treatments, or at least we didn't have good clinical trials, and we didn't really have good controlled evidence to support what we were trying to do. So I give this from the 1890s, which is when ulcerative colitis appears to have emerged or at least been clarified, all the way up until the 1990s, and I'll tell you why that is. This is actually a picture Dr. Kersner gave me years ago, and it was only when I was preparing this talk that I found him describe it in writing. He had often described it to me, but he said, "My interest in IBD began in January 1936 when I undertook the care of a 40-year-old emaciated woman who was brought to the University of Chicago in desperate condition with severe ulcerative colitis, a virtually unknown illness at the time. She rapidly succumbed to the disease," and you can clearly see why. That's Dr. Kersner on his 102nd birthday with my other mentor, Dr. Hanauer, who's younger than 102. Oh, you got that on camera, didn't you? And then if you look back at some of the early theories about what caused IBD, you can see a variety of things. And I know this is a small print, but I've highlighted a few for you. The idea that this is an infection or alteration of the microbiome or the flora in the gut or some infectious type of organism comes up over and over and over again. People kept thinking this must be an infection, we don't know what it is, or this must be triggered by an infection, or there's some imbalance of the organisms. And every once in a while, people would put their money on a single organism. They would say, this is what it is, we just can't quite treat it properly, or we can't find it properly. And so, depending on what the theory was, there were a variety of different therapies that people tried. Some of them are quite extreme. And I'm sure some here in the room understand and remember when they thought this was a psychogenic illness, and they would hospitalize people for months at a time and prevent them from seeing family members and enroll them in group therapy sessions to treat their IBD. There are even stories of frontal lobotomies to treat IBD. So when we look at the flora in the gut, there actually have been many attempts to modify the flora, not just for IBD, but there have been some descriptions of doing that, of course, for C. diff. And most recently, this has become of great interest. There are repeated descriptions of large case series or larger case series now from groups around the country that are saying if you do a fecal transplantation, you can cure C. diff with one treatment. And in fact, that appears to be true based on what's being published. But this goes back a long way, and people have tried this for a variety of different ailments and with a variety of different techniques that I'll summarize for you in a bit. When we get to 1990, I picked 1990 because it was when the first drug was FDA approved for ulcerative colitis that was specifically studied in a controlled trial. I call this the era of discovery and evidence, the era of endoscopy, of course, and the era of the internet. And that goes all the way up until right now. And I didn't pick right now because I was giving a talk right now. In 2012, I think there are some very important specific things happening in therapies and in the science of IBD that are going to lead to more and more individualized care, and that's the next era. But when we focus on this era, you can see a variety of things that we start thinking about. Because we can control the disease, because we have better surgical techniques, we can now start thinking about longer-term management. So that's when you start appreciating how do you change outcomes. That's when you start appreciating the importance of quality of life. And that's also when you start to appreciate that if you have the ability to modify the disease and you have some good therapies, you can start focusing on patient choice. Like, when do you want to take your medicine? How do you want to get it delivered? And when we start talking about things like that, you know you must have made some progress in your disease management. Now, despite that, I want to point out to you that there's a huge gap in what we need to do. So if you're a patient suffering from ulcerative colitis, the glass can seem half empty. And the reality is that although many of our patients do quite well and most patients have milder disease, we still have an unacceptable number of patients who have either progressive fulminant disease or lose response to therapies and end up needing surgery. So we're still talking about a 25 to 30% colectomy rate, depending on whose studies you look at. And even this, which is now considered to be quite old in terms of looking at this, you have to accept the fact that in more recent studies and analyses, our colectomy rate hasn't changed very much. So at a minimum, there's a group of patients that end up with surgery for their disease that we'd like to try and identify and understand better. And then there's a whole group of people who live with sort of unacceptable disease control. Now, just to emphasize what happened during this era of controlled evidence and emerging therapies, we can think about how we try to treat ulcerative colitis, which I would argue is actually not a very efficient strategy. It's based on you try the therapy that might be less expensive and safer first, and if it works, you're happy and you keep going. And if it doesn't, you step up to the next level. This hasn't changed in many years. We haven't really gotten to the point quite yet where we're using targeted therapies or where we have a good idea to say, with this particular patient, I need to start with this therapy, either because of better benefit or, more importantly, in some ways, less adverse events. We're not there quite yet, so we're still going up this staircase, and that's a problem in our management as well. If we look across all of our therapies very quickly, you can see the obvious gaps. And I'll point out to you, first of all, that there's the very important observation that the y-axis on some of these graphs doesn't go to 100. So you have to keep in mind that there is actually a huge gap of patients who don't respond or don't achieve remission or don't maintain remission. Starting with induction of remission, I'm going to focus more on ulcerative colitis for much of my presentation today. But you can appreciate that even with drugs that we think are effective for mild to moderate disease, we're only achieving remission in a fraction of those. If you look at actually across the 5 ASA studies compared to placebo, you can see that the gap is substantial. Maybe half our patients don't respond. So having a greater understanding of why people don't respond as primary therapies to some of these strategies is really an important point and something that we should be focusing more on, and that's some of our other research that we're doing. Even if you look at steroids which we hate to use long term and our patients often hate getting at all, you can appreciate that first of all there's quite sparse evidence for how steroids work in UC, even though we use them. And there's a huge gap in terms of using steroids to induce remission or at least response in ulcerative colitis. So when you focus on the IV steroids, the sicker patients, this nice meta-analysis demonstrates the number of patients who still ended up with colectomy. And you could argue that once you give steroids, that's already one of the outcomes you should be thinking carefully about and preparing for, but also the number of patients who just don't respond to it at all. And there's a variety of potential genetic, disease-related, and other factors that could explain that. Once a patient does get steroids, though, what do you do with them afterwards is another gap in our understanding and need for better treatments. So in this particular analysis, they were looking at taking patients who responded to steroids who had ulcerative colitis and randomizing them to a strain of E. coli compared to mesalazine. You can see they both did equally poorly. And so there's a huge problem in terms of maintenance of control once you have patients who respond. If you look at 6-MP and azathioprine, the standard approach after steroid use in many of our patients and sort of our traditional approach in Crohn's as well, you can see that this is not an effective strategy in a majority of patients. So I'm here today to point out to you that despite our advances, there's still this very large gap in understanding the disease and understanding how to control it. And so when I give talks to patients, I of course don't say this. We're focused on trying to make sure people are well and getting good options for all of them. But this really is, I think, an urgent need for better treatments and better understanding for the disease. Even if we look at infliximab, which some would argue that has made the most difference, certainly in Crohn's, and maybe there's some ways that it has in ulcerative colitis, or at least has the best evidence so far, you can see that we're still only talking about a 35 to 39% remission rate in 2 large trials of outpatients. So lots of room for improvement. So aside from the fact that we can't cure this disease medically yet, we also know that even when patients respond to therapy, they then lose response over time. We also understand that there are some safety profiles that are real that we have concerns about, and then there are the perceived safety concerns that our patients have, which are just as real to them and make it difficult to prescribe therapies or at least to keep patients on therapies. Another issue that's really important for all of us in any healthcare industry and specialty is really access. Our patients have inequitable access to therapies and inequitable access to options for their care. So we have some big problems here. I tell you all that to tell you this, which is that we're getting to a point now where we're hopeful that we're going to start individualizing our care. And we're getting some very good ways to do that, and we're understanding this more and more. First of all, IBD is being reclassified. So it'll be most important to know about the reclassification when it actually influences treatment choices and an understanding of prognosis. But between molecular, immunologic, and biologic classification systems, we are going to start stratifying patients and choosing therapies based on likelihood of response. We're also starting to use targeted therapies. So in February was the announcement of a phase 3 trial result that has a therapy that is delivered IV, but it's only targeting receptors on the gut. So the idea that we're going to target the gut specifically is a very nice idea and a good concept if you believe that this is a gut-specific disease and not a systemic immune problem that's targeting the bowel, which is a different discussion. But we're getting there. We have certainly better surgical techniques, and we're grateful for those, and we're obviously focusing not only on quality of life, but quality of care. So there's a lot that we're doing, but the rest of my presentation is going to focus on one potential segment of understanding the disease, and that's focusing on what I call the final frontier, which is the microbiome as it relates to IBD and more specifically to ulcerative colitis. So I hope you can appreciate this graph. This is actually from a recent publication in gastroenterology that demonstrates to you what's being observed around the world. There's some bias in this as to where studies have actually been carefully performed as opposed to anecdote. But based on evidence, you can see the difference between this graph of the worldwide incidence of IBD and in the era before 1960 compared to the time between 1960 and 1979 where things are starting to get more obvious and maybe more studied. And then the most recent era where you're seeing actually quite a rise in some obvious areas. This is just ulcerative colitis. The paper nicely does the same for Crohn's, but I'm not going to show you those graphs today. But I'll demonstrate to you that in some places it's clear that IBD is on the rise compared to previous because they have really good databases and they've done nice studies. And there's other places where it just never was studied before, but when they did, they realized, gosh, in New Zealand we actually have quite a high rate of IBD, and we didn't previously document that. So it's appearing on the map. We keep hearing about it rising dramatically in China as well. They just haven't done a formal epidemiologic study yet, so they're still just sort of collecting the gestalt of what's going on, and I think that there's some very important studies going on now that'll help to tell us more about that. So why might IBD be rising around the world, and specifically ulcerative colitis? Well, there's a variety of theories that everyone in the room has heard something about. Whether it's related to just better studies and documentation of what's actually been there all along, which most of us think is probably not the case, or whether it's related to the hygiene hypothesis, infection, some kind of westernized diet influence, which now we acknowledge is that the westernized diet changes the gut flora, or what's been termed, and I think came from here, the microbiome hypothesis. You can think about all of these things as being related to the gut flora, either way you spin it. So we have to start thinking carefully about how is the gut flora changing and why is the immune system responding to it this way that leads to these inflammatory bowel diseases. So there's a variety of ways to think about therapy and using the microbiome as a way to treat it, because if we acknowledge that there might be a difference in the flora, then we'd like to know how we might restore it or do something to adjust the immune response to it to develop treatments. We'll first start with our current understanding of what's going on in IBD. And this is a simplistic view, but it actually applies to every patient. You can think about some patients being more genetic than others. But a genetically susceptible host is exposed to something in the environment, which may be even just a change in the flora, that leads to an unregulated immune response. So this is not an autoimmune disease, as you know. This is not an allergic response, as far as we understand. This is really a loss of control. So that's how I try to explain it to patients and try to help them understand why we use immune suppression as a treatment strategy. But some patients are clearly influenced more by the environmental factors, like the ex-smoker at age 44 who gets ulcerative colitis. Others are clearly more genetic in terms of when they develop their disease at a young age or even infancy, and appreciating that those distinct differences clearly demonstrate some different pathogenetic mechanisms and pathways will help us understand more about how we choose different therapies. There's a variety of evidence, both positive and negative, about the role of gut flora in IBD. There are the clear clinical observations related to the onset of IBD after antibiotic exposure. We've all seen patients who tell us about their antibiotic exposure, and now there's the emerging studies that demonstrate that childhood antibiotic exposure increases the likelihood of developing IBD later. So we know that there may be a relationship there. There are some really nice animal models in germ-free facilities where the animals don't develop IBD until they're exposed to the organisms, and that's clear, although that doesn't translate as well to humans where we're clearly not in a germ-free environment. There's all the serological markers of IBD that reflect leaky guts, and now the emerging understanding that the microbiome has a direct influence, and even diet has an influence on how leaky your gut may be. We also know that family members of patients with IBD have leaky guts, so they may have some susceptibility, and then there's something else in the person who gets the IBD that turns it on. We certainly have appreciated for many years now that if you divert the fecal stream, you can put Crohn's into remission. And one of the ways that's become explained is that you're essentially taking the stream away from the bacteria that live in the colon. And when you do so, you essentially turn off the immune response. We know a lot about post-op recurrence of Crohn's and how it seems to be related to bacterial exposure, or that you can use antibiotics, at least short-term, to prevent recurrence after Crohn's surgery with primary anastomoses. And then perhaps a separate inflammatory bowel disorder, pouchitis, is responsive to antibiotics. And there's lots of actually microbiome studies going on related to that right now. There's inconsistent evidence related to probiotics, however, And even though there's been some exciting and interesting studies performed, the reality is that a probiotic approach to managing or modulating the gut flora hasn't really demonstrated success in repeated trials and an ability to use it in our clinical practice, or at least in most patients. There's a variety of challenges that we have to face if we're going to study this in our patients. Number one is we have to have a classification system. We have to first know, how do you describe somebody's microbiome? So what do you actually need to do to make this translatable and understandable? And that's probably the first priority. Number two is understanding what happens to an individual over time. So if I was able to classify your microbiome in your gut today, is it going to be the same tomorrow? Or will it be the same after dinner tonight? Or what happens to it after you get an antibiotic? Or what happens to it when you have IBD? So there's some very important changes that need to be documented and better understood. Third would be understanding disease-related heterogeneity. So what is the difference in one patient with colitis compared to another? How do patients with colitis compare to patients with Crohn's? And of course, how do these compare to patients who are either healthy controls or people with other gut disorders like irritable bowel? What do we do in terms of developing these trials? And that's where we're going to spend a little more time talking about in a moment. And what are the valid endpoints of such trials? Should we rely on clinical endpoints, or do we need to include such translational and important microbiota-related endpoints? How do we deliver the change agents? So we have to do this effectively. We don't want to have a theory about how to do all this only to deliver something that doesn't actually change the gut flora effectively. So we have to understand what we need to actually do. Is this going to be using antibiotics or probiotics or prebiotics, or what is euphemistically called microbiota extracts, which can be stool or extracts from stool? We have to standardize our interventions and understand how to do that. We have to address safety issues in patients and in the population, which I'm going to focus on today. And we have to understand changes in the microbiome. Are they actually cause or effect, or both? Now, all of these questions came up when we started thinking about how we wanted to actually study this in our IBD population. And we've tried to address some of them based on what others have done very nicely, and others we think we need to still do more work in. So I'll try to summarize a few things. The first is that actually there's been remarkable advances in our ability to classify what's going on. So the primary technology that most have been using recently is the 16S RNA technology, which helps us classify the general microbial communities in the gut, even if we can't culture many of the organisms. So this enables us to do a variety of different techniques to say not only what's there, but what might it be doing, and comparing it to people who have different disorders in their gut, or even in individuals over time. So this was a really important priority, and there's still good work going on here. But until this is simplified enough that we can use it in clinical practice, it's going to be still a very difficult technique for us to apply to our clinical trials as well. It requires, obviously, access to the bioinformatics and interpretation of these results and this information, which is at a level that is much more than anything we've looked at before. Now, there is some really nice work now that demonstrates two things. Number one is that when you look at IBD, it's different than healthy controls. And number two is that the gut microbiome does have some changes over time, from birth to weaning to elderly patients. And you can appreciate the differences. There are some organisms and general species that are increased in IBD compared to controls, and then there are some that are decreased in IBD compared to a healthy control population. And so when we start looking at this, this is one of the major clues in terms of general organisms and strategies that we might want to focus on. So if these are generally decreased, you can understand why some people have focused on using lactobacillus probiotics or bifidobacter probiotics as a strategy to try to treat the disease. But we're skipping some steps in the middle to actually understand whether that's important or not, and whether that's cause or effect. There have been some nice studies, that, albeit in small patients, that have started to give us some clues that not only do patients with IBD have differences in their microbiota compared to healthy controls, whether you're actively inflamed or in remission, your microbiota will vary. So in this small study, but very important one, you can appreciate that Crohn's disease that's inflamed, which is what the CDI stands for, compared to UC that's normal, or UC that's inflamed, or Crohn's that's normal, and then healthy individuals, has variations. So the inflamed gut has clearly some change in the gut microbiota. When you get them into remission, that goes back to normal. Now, whether that's because the gut flora are part of that process, or whether because the gut inflammation is turned off, the gut flora then normalize, hasn't been fully understood yet. We also understand that IBD patients appear to have more variations in their gut flora over time, which is related to that last study. In other words, if you look at an IBD patient over time, it really does matter when you sample them, because depending on how severe their disease is, depending on what therapies they've been exposed to, and depending on how inflamed they are at the time you do it, you're going to get different results. So you can take some general ideas about what's going on in them, but in those patients, especially compared to healthy controls, there's going to be some variation that needs to be accounted in any studies that try to look at these outcomes. Very important. So a single point in time may be insufficient to actually understand what's going on. What is the therapeutic potential? What could we do, and how would we design some studies? Well, we can start by looking over the probiotic therapies, and I'm grateful to Richard Fedorik, who's done some great work in this area, that shared his slide with me here. So I'm only going to show you this one example of probiotics, but we know they've been studied in a variety of other disease-related states in IBD. In ulcerative colitis, just looking at the success of probiotics in inducing remission, whether you're looking at the E. coli Nissl strains, you can appreciate that they did not have statistical significance, or at least was borderline statistically significant in the small trials that have been done. There's been great interest in the VSL3 product in a variety of different studies, and some of them have actually demonstrated some success. And so we've been enthusiastic about that possibility without actually understanding what's actually happening. But then again, we don't understand what's happening with some of our other therapies. So although this isn't a common strategy for treating mild ulcerative colitis, it's still worth acknowledging the evidence that suggests it may work. And then there's some other interesting studies looking at fermented milk, or even some other combinations of that, where you can see that there was potentially some statistical significance. So as much as we don't use probiotics as a traditional strategy, and we may criticize the size of the studies or our lack of ability to reproduce them in some ways, you have to acknowledge that there is some benefit in some patients. If you look at maintenance of remission, even as an additive therapy on top of 5-ASA, and certainly in probiotics, which is a whole other lecture, we understand the role, in pouchitis, we understand the role of probiotics. So when we talk about now designing these trials and thinking about all this, let's think about some of the basic ethical principles we should keep in mind. The first one is beneficence versus non-maleficence. So of course we want the benefit, we don't want the harm. The second is understanding patients' right to choose and some of their interest in what they're going to receive as therapies versus what we consider to be our current model, which is shared decision making. And the third one is really about justice, or what I'd like to term more as resource allocation. So one of the things we have to keep in mind is also expense and cost of therapies as we go forward. So biological therapies can be very expensive, and although we're developing some very important biological therapies for our use in IBD, the reality is that the cost of them is currently going to restrict our ability to use them in most patients, or at least many patients. So why don't we take a sequential assessment of our approach. If we were going to do this in the traditional way, where we believe this is an infection or some microbe-related illness, we would start with our theory, which I've sort of laid out for you, about the role of the microbiota in the pathogenesis of IBD. We might identify an organism or series of organisms that we think are causing or specifically and directly related to the disease itself. We then want to try to prove that they actually adhere to Koch's postulates. So everyone in the room who's taken microbiology remembers what that is. We need to isolate the organism, we need to document its association with the disease, we need to demonstrate improvement when it's treated, and then we have to demonstrate that you recur the disease when you actually reinfect the individuals. And then after that, we develop targeted interventions. That's the traditional strategy. What people have said is, well, the gut flora is so complicated and we don't have time, we want to try to skip ahead. There's been a single organism that, if you go back in the history, this is back in Dr. Kersner's times, these were actually a surgeon and a couple other colleagues at the University of Chicago who had this organism. At the time they called it Bacterium necrophorum. Now it's a fusobacterium that's still around. People are still interested in it. This anaerobic organism, non-sporulating, has been associated with ulcerative colitis. They thought this is the one. This is causing ulcerative colitis, going back to 1936. They isolated it, they cultured it, and then they tried to cause the disease in animals, and it sort of stopped there. I tried to track the history of all these publications, and I asked Dr. Kersner about it. And now it's reemerging. There's discussions and interest in Japan of this organism, and people are starting to say, well, what did we actually do there, and what are we missing? So that might get to a single organism as a causative effect. And there's some interesting animal models in which specific organisms cause specific phenotypes of colitis in mice, which is an interesting idea too. In the absence of all that, though, you're left without the ability to really understand how you're going to do direct treatments. So you have your theory, but instead of developing a specific organism, you skip the middle step and you develop some kind of untargeted therapy, which is the idea of doing a fecal transplant. You're basically saying there's something good in people's healthy stool. We're going to put this all into somebody who's sick and see what happens. Now, there's potential problems with that, as you can imagine. And then based on whether it works or not, you then go back and say, well, why did it work? And so this is a different strategy. And that's where some of this is coming from. So the methodology of this, and this is not new, this has been around for a while, but the term is new. So I'll encourage you to start using this term, which has been accepted and now widely published, which is fecal microbiota transplantation. Some have called it the ultimate probiotic. I think you could argue with that term. The idea is you identify a healthy donor, I put that in quotes. You collect stool from them, you screen the donor and the stool for infections, you transfer the stool, or at least the result of your preparation by enema, colonoscopy, or nasogastric tube to the patient. And in C. diff, in multiple series now, as I mentioned, this is an extremely effective strategy for eradicating recurrent or refractory C. diff. In fact, it's greater than 90%, maybe as high as 95% in achieving control and eradication of the disease, even with longer-term follow-up that's just been published. In ulcerative colitis, we have a very limited data set. There's this paper that first piqued our interest from Tom Barodi, who I've now met and discussed this with multiple times. And he's been very collegial about this and very interested in this topic for a long time. He published this series of 6 patients with UC who had refractory disease and in whom he prepared fecal transplant with a single colonoscopy and delivered it, as enemas, and then had daily treatment subsequently for 5 days. And these are those 6 patients and what was published and described to these patients with their follow-up. And you can see that between 1 and as long as 13 years of follow-up, these patients' histologic follow-up demonstrated normal histology of these patients with refractory UC. So there was a lot of enthusiasm about this. What are we missing? Why aren't we doing it yet? And subsequently, some people have tried this. And I know Dr. Barodi has done this in many more patients. His enthusiasm, I think, is somewhat waned based on his own experience, although I don't want to misquote him. He's acknowledged that UC is clearly different than C. diff. Many of his patients require multiple treatments, and the success rate is not what this uncontrolled series suggests. There are a variety of problems we have to think about. If you were going to design a trial like this, as easy as it sounds to just screen someone and stick stool into another person, the reality is that we have to think carefully about who we would include. Ulcerative colitis or Crohn's. If we use Crohn's, should we use Crohn's of the colon only, or is it the ileum too? Adults or children? You could say, oh, well, we should always start doing clinical trials in adults first. There's some issues related to consent. But you could also argue that it might be kids that have more changeable microbiota or may have a shorter disease duration that might lead to a better response. What about severity of disease? How do you classify that? Are you going to let patients with long-standing refractory disease have this type of treatment? Or should we really be targeting people with shorter duration of disease? And then understanding how all the therapies they've been on before they get into a trial. Do we want to take the patient who's not responded to every therapy, who's been immune suppressed, who's received multiple courses of antibiotics, and do this to them? Or should we really try to be very selective about who we study this in so we don't get too confused with our results? We've started to explore some of these ethical issues, and we published just recently the results of a focus group where we looked at our patients who had UC, and we did 2 separate groups of parents of kids with UC. This was done by Stacy Kahn, who's a wonderful pediatric colleague and interested in IBD and also has done ethics training. And in general, as you might not be surprised, patients think this is very natural. This is a natural treatment, and therefore it must be safe. That was the general theme that came out. It was perceived that it must be safer than the traditional therapies, and they want it now. As soon as you can give it to me, I'm interested in getting it. And it was interesting that despite some of the patients being in stable remission on their other therapies, they would still be interested in this. Now, the way you do focus group analysis actually requires transcription and developing themes and making sure that you're exhausting those themes before you move on. But we broke it down into some different domains, and there are a variety of different things that came up that help us design the clinical trials and make sure we're addressing our ethical concerns and our IRB's concerns. But just to summarize a couple of the things people said in these focus groups, One was basically a patient who sounds more desperate. I'm ready to try anything, okay? And the other is a general theme that came out a lot, which was they thought this was natural. This must be natural. So taking someone else's stool and putting it in your body is natural, right? Because the body makes stool, so it must be natural. Well, there's some concerns to be thought of about that. Based on our focus groups, we designed a follow-up survey, which Stacy's presenting at DDW. And so essentially the same, but one of the things that we focused in on as well is who would you want to be your donor? Would you rather have a family member, or would you rather have somebody the doctor told you was clean and appropriate? And that was split, actually. We thought more people would want family members, but it ultimately seems that they either trust their doctors at the U of C, or they actually do have some potential hesitations about just having family members do this for them. And there's some biological reasons to think about that. We know that people living together tend to have microbiota that are similar. We know that family members may have microbiota that are similar, and maybe there's other genetic factors that would make your response to a family member's microbiota less good. But we're still trying to understand that a little bit more. Now, I just created this cartoon to demonstrate one of the things we observe in clinical practice, right? So patients like oral therapies. They don't like us to give them injection therapies. They like worms, because probably for the same reason. They're very interested in that idea. And they seem to really be excited about stool. So depending on the yuck factor, I think that goes along with the patient's perception that this might work. So if they're able to endure something that on the surface sounds quite awful, they're probably more likely to respond to it. And that seems to be what's coming out of our surveys and our focus groups. So let's move on, though, to some of the real issues here regarding protection of human subjects. The first argument I'm going to make is that desperation is never an acceptable way to get people into clinical trials. And since this paper was EPUB'd, not even in a full printed form, when we published our focus group analysis, Stacy and I were inundated with emails from patients all over the world asking us to do fecal transplants on them. We didn't say we were doing them yet. We said we did focus groups. And I was getting emails every day or every other day from patients around the world asking if we could please do this to them. And they would summarize their whole clinical history, all the therapies they'd been on, including all the complementary therapies they'd already tried, and they would end with something like, and I know this will cure me, and they want this therapy. So I would start by saying that we have to be especially careful about people who view this as desperation, and that's important. A second issue that comes up often is I don't think people should be in clinical trials just because they don't have insurance, and that comes up all the time, and I think we need to be extra careful about protecting those individuals as well. Our clinical trials of FMT need to protect these individuals, and there need to be some safeguards in place. Our IRB has made that clear, and we agree with them. So first of all, patients need to understand the limitations and the lack of information that we currently have, and they also need to know what their alternatives are. And we have to protect donors too, which is a separate issue that I'll bring up in a moment. So we've actually proposed more of a phased study design for this. Rather than continuing to do case series and publish uncontrolled experiences, we really think that we should try to approach this in a more methodical way. Phase 1, which is where we are, is what we want to be safety, feasibility, and some specific translational outcomes demonstrating that there's actually something measurable that's being changed in these individuals. Phase 2 would be refining the technique, dose ranging, and longer-term follow-up and efficacy. Dose ranging meaning, of course, is this something where we can do a single colonoscopy? Do we need to do multiple enemas? And what are we going to do? And of course, phase 3 should include controlled trials. This should not be different than anything else we do with chemicals or biological therapies. And we should not get carried away with patients' willingness to do it, or the fact that we think that it's an enthusiastic approach to let us skip those steps. In addition, the FDA has stepped in. So not only for our study, and our IRB asked us to talk to the FDA, but for the people doing this in C. diff. The FDA is now requiring an investigational new drug application. So even though this isn't a drug, and even though we don't plan to sell it across state borders, the reality is that they want to make sure that they understand what this is and what it might be, and that process is quite involved, as you might expect. The C. diff folks have led the way in this, and they've been very gracious in sharing their work. And we're doing the same now for ulcerative colitis before we start our trial. So that's going to take some time, but we're well on our way. The second issue is really management of the fecal material. So within your own institution, you have to know who needs to be involved. So we have a data safety monitoring board, but we included infectious disease. We included our safety folks from the laboratories, from microbiology, and we had to figure out where and how and really what were we going to do to prepare this. In previous descriptions and on the internet, people just buy a blender at home and they bring it in pre-emulsified and they hand it off to the doctor. We're not really encouraging that approach, and that's not what we're going to do. So these are some issues you have to think about. What about donors? So most people say, oh, donors, it's easy. What's the risk to a donor? It's not that hard. Give them a newspaper and then you collect. So the reality is that we have to think about this in a variety of ways. Number one is, should we treat this like organ transplants? So you have to remember that living organ donors are people who have an opt-out. So when they're asked to donate to a family member, the opt-out is that if there's some reason that they really don't want to donate, but they can't say it or they feel coerced otherwise, we as the physicians and the healthcare team are able to say you're not a match, and therefore we can't do it. And there's no questions asked after that. It's a very easy opt-out. If you were going to screen donors for a variety of diseases and then reveal whether or not they're clean for you to be able to do the transplant based on their donor status, there's not an easy opt-out here. You can't say you're not a match with some very specific exceptions. So our IRB actually had a whole ethics meeting about who the donors should be, which was very interesting to discuss. And you can imagine some of the comments that were made and volunteers that came up. But the reality is that they actually favored us thinking more about this like blood transfusions, which is that maybe there should be screened individuals in our early trials who are clean and who we know that, all about their history, who are not known to the recipients. And then the question becomes, well, who's the ideal donor from a biologic point of view? And we don't necessarily know that. For our clinical trials, the screening will come out of our grants. But in the reality of the next step, who would pay for screening of donors when you have patients who need these therapies, if it's actually effective? And that's another big question for us. Remember that IBD is not C. diff. So the fact that it works so well in C. diff does not at all mean it's going to work as well in UC or in Crohn's. And in fact, our colleagues who are experimenting with this in Europe and even in the US, one or two case anecdotes are now trying to tell us that more and more. This is clearly different. The other thing to know is that although this is an important paper that was just published by Larry Brandt and the rest of the C. diff group that are studying this, looking at longer-term follow-up in their patients, very impressive results. No doubt about it that this is important. I do want to point out to you, though, that there was one patient in their series who had Crohn's who died of subsequent sepsis, and it wasn't directly related to the transplant. It was 5 months later. And so they said it was unlikely due to their fecal transplant. But I have to include that as something that was downstream, that it happened to a patient. And I'm sure there are other reasons a patient could have sepsis with Crohn's. There was also this emergence, and this is early, but there were 4 interesting new diseases that occurred in the people who got treated for C. diff with this strategy. And they didn't know what to make of it, but they mentioned it in their paper. One patient had ITP, one got RA subsequently, one had a peripheral neuropathy, and someone else was subsequently diagnosed with Sjogren's. Complicated patients, who gets refractory and recurrent C. diff, the sickest patients with lots of problems. So I'm just pointing out to you that this is not quite as simple as treating and not thinking about downstream effects. There's a lot going on in the gut organisms, and we have to think carefully about what we're going to do to our patients as we do this. So let me show you in 2 slides what we're planning. Our phase 1 study has these specific aims. We want to establish the safety and efficacy of fecal bacteriotherapy in mild to moderate UC. Not doing Crohn's, we're not doing severe disease. We're going to characterize the microbiota of the healthy donors and the UC patients and determine what happens to the microbiota. So the clinical endpoint is actually not the primary purpose of our phase 1 trial. And then we're going to look at some of the ethical and safety concerns related to this. We're planning 15 patients, and this is based on what we think we need to demonstrate feasibility and safety, at least initially. And we're going to let them fail 5 ASAs, specific lower doses of steroids, or stable doses of thiopurines. These are not patients who are hospitalized, they're not severe disease, and they can't be on biologics or higher dose steroids. Donors will be unrelated in our study design, obviously no IBD in them or their family members. And we're going to screen for the variety of different things that you might think about, and based on our discussions. We're not sure about CMV and HTLV yet, but that's going on with our infectious disease folks, and we're still working through that. We're going to use a colonoscopy delivery system, which is similar to what's been described mostly in C. diff, a single colonoscopy where it's sprayed in. And we're going to then do our 16S RNA assessments to see if we can actually change the flora compared to the donor and compared to baseline. And then we'll do clinical endoscopic and histologic assessments at specific time points as we follow these people out. So that's what we have designed, and pending the IND, we're ready to get started. But that's a big pending, so we're going to keep waiting for that and work through the FDA issues. So I'm going to stop there. I've certainly covered a lot of ground here in terms of the history of IBD, why we think this may be an important element and a major advance in our field, but what we need to be thinking about if we're going to translate some of this important work into clinical trials. There's clearly some heterogeneity issues as well as some very important safety and ethical concerns. We have a very busy and big IBD center, and many, if not most, of these individuals are directly involved in all the work that we're doing related to this. And there's a variety of different funding mechanisms that are supporting what we're doing. So I want to acknowledge all my colleagues and all their dedicated work to help us with all of this. So I want to, again, just thank you very much for letting me come visit and see your very impressive center and being such gracious hosts. And I'll stop there and maybe take some questions from the audience. Thank you. Yeah.
This was actually described back in the 80s in case reports and was experimented with at this institution, believe it or not, and also at University of Colorado. My understanding from those original experiments, it was a handful of patients again, was that it died due to lack of efficacy. Are you aware of that old data?
We've seen some of that, and we've seen even data from the 60s too, which are all just these single cases or these
Like
limited cases.
Right.
Anecdotally, there's been a small series done in Austria that I know of now. And most of the time when you hear about those patients, these were people who were 1 foot into the operating room where they decided to do something. Not always. There's actually a controlled trial going on right now in Europe. And their targeted goal is to recruit 60 patients. I think they have 20 that are already done. And so I can't comment on what their results are so far, but I think that the observation from the 80s may hold true as we start to move into this disease state. Yeah. Yeah, John.
Microbiota is highly influenced by the diet.
Mm-hmm.
So in the trial that you are doing, are you controlling the diet?
That's a very important question, so thanks for bringing it up. We are controlling a variety of things, including antibiotic exposure, but diet is also something that we're collecting. We are not sure how to control it, what we would do to do that adequately in 15 patients. But clearly that's an important variable that we have to think carefully about. And we've been talking extensively about how we're going to do that. It's actually why we actually had 2 nutrition dietitian people helping us with this, and we're going to collect food logs and try to make sure that there's nothing that's an outlier, but it's a very important issue.
I frequently worry about microbiota of the stool not necessarily representing what's attached to the mucosa. And we don't really know which is more important.
Right.
How are you going to measure the changes in the microbiota?
Well, we're planning to use biopsies and follow up flex SIGs with more biopsies. So although it's more convenient to collect the stool specimens, we think that probably the mucosally adherent organisms and doing it that way consistently is going to be more informative. But then the question becomes really, well, where are we looking? If we're doing a flex SIG in the distal colon, that's important, but what goes on in the distal colon doesn't look like the proximal colon either. So there are many feasibility issues about doing this as extensively as we'd like. We'd like to get our foot in the door and actually demonstrate we can do it. And then thinking about how we're going to be more careful with some of these other assessments later is going to be part of it, no doubt. Grace.
John Kale in our division and some of our infectious disease doctors have written an IRB for C. diff treatment, and we're ready to get started. It's interesting, I didn't know that there was an IND, though. I don't know if they know about that.
They do as of yesterday.
I was going to say, that's brand new news to me.
Well, thankfully, Colleen Kelly from Brown has led that for C. diff. She's been terrific, and I'm going to make sure that they're circled in on it, but they're well along the way. So the FDA already responded to them and has some questions, and they already submitted their clarifications, and that's looking like it's going to be.
Well, what my question is that our microbiology department refused to help us. Essentially said, absolutely not, we're not doing it. So we were going to prepare the stool ourselves in our endoscopy unit. Well, how are you managing it at your center? That's
Well, we didn't ask our microbiology department to prepare the stool. They
we
probably
did.
would have had the same response as
Yeah,
yours.
and they said no.
They've been collaborating in terms of helping us decide how to screen the stool and what we're going to do from that perspective. But we're going to use one of our appropriate labs for our clinical research that has a hood and prepare it in those settings by our own technicians who are all going to get raises. And then hopefully it'll go smoothly from there. Yeah. Hi.
So I noticed that you're planning on using the TLFOP and the 498.
Yep.
Why not 494 viruses?
Well, actually, since this was originally designed and our grants were submitted, we've modified that, and we're hoping to secure some additional funding, and Gene wants to do that. And part of what's in this larger grant is storing additional samples and making sure we have the ability to look at other things later. So we will undoubtedly be doing that too. There's no reason we're not doing it. It was just when we started this whole process in designing this trial and going to our ethics department or our microbiology department and coming up with a DSMB and going through the research and literature and meeting in Australia with a couple people. All these things took us over a year to do. So we think we're doing it the right way so far. Not that people aren't doing it the right way in other places, but we're comfortable and our IRB is comfortable. And they would have signed off if it wasn't for the FDA. So we would have gotten started by now otherwise.
And just a real quick question. Comparison between enemas versus colonoscopy delivery. I haven't looked at the literature, but I was told that it was very similar. Your opinion?
Well, so in C. diff at least, it seems like it might be similar, which you might be surprised by because C. diff can affect the whole colon and sometimes more preferentially the right in some patients. But our general concept would be that UC patients especially have difficulty holding enemas. And so we thought that, and what's been described for the UC patients is that when you spray, most of what you do is in the proximal colon, and they actually have the patient use Imodium afterwards for retention, and then you let the rest take its course, and that seems to be effective. But people are sort of making this up as they go along. The other issue is, do you prep them before their colonoscopy? And the answer appears to be yes, we are going to prep our patients. And then you have to understand whether the prep itself is a treatment that might affect your results. And that's important. So we're going back and forth about that. And that's another reason why, frankly, we're going to have controlled trials if people are still enthusiastic about this as it moves forward. Because unless you do something to control for these things, you're not going to know what you're really doing.
Thank you all for coming.